using Punnett squares to analyze a pedigree chart. Here we have an example of a pedigree chart. Some members of a population of rats have an autosomal recessive condition called albinism. Rats with albinism lack pigment in their hair, skin, and eyes. The pedigree chart shown is from a sample rat population with an incidence of albinism. We are going to examine the number of rats that would have genotypes which are homozygous recessive, homozygous dominant, or heterozygous. Keep in mind the trait we are looking at is recessive. To begin, write in the information that you are sure about. Remember, circles are female and squares represent males. A black square or circle must have 2 recessive genes because the trait is being shown. A white square or circle may have 2 dominant genes or 1 dominant gene. Using this information, let's fill in what we know. The female in row 1 has at least 1 dominant allele. As it is white, the trait is not being shown. We do not know what the other allele is at this point. The male in row 1 must be homozygous recessive, as the recessive trait we are examining is being shown. We can use the same logic to fill in the rest of rows 2 and row 3. We are missing the second letter for many species in the pedigree chart, and we must use Punnett squares to analyze what they could be. To begin, examine the possibility of the female in row 1 being homozygous dominant, 2 capital letter A's. Construct a Punnett square using this assumption. Are the offspring from this Punnett square a possible combination giving the pedigree chart? No. The offspring from this cross are all heterozygous and will have the dominant trait showing. This does not match with the information in the pedigree chart, as we see some of the offspring are homozygous recessive. So this is wrong. Next, try the possibility of the female in row 1 being heterozygous. Construct a Punnett square using this assumption, and we will see that the offspring from this combination match what we are given in the pedigree chart. Some are showing the recessive trait and some are showing the dominant trait. We can now fill in the missing information. We have now determined the alleles of all of the offspring of our original cross, these 4. However, we have not addressed the last male in row 2. This individual is not an offspring of the original cross, it is a separate individual which is now being crossed with one of the offspring. How did we determine the second allele? Well, we did the reverse of the previous process, by analyzing the offspring of this cross. We see here that the female in row 2 is homozygous recessive, meaning it got 1 recessive allele from the mother and 1 recessive allele from the father. Because of this information, we know that it got a recessive allele from the father, and so this one has to be heterozygous. Perform the same cross of these 2 individuals, and we can determine what the genotype is of our male in row 3. All the missing information has now been determined. So we can go through the pedigree chart and count up how many are homozygous recessive, that would be 4, homozygous dominant, 0, and heterozygous, 5.